Good morning. It gives me great pleasure to introduce Dr. Una Fairbrother. Thank you for coming to do my talk.
It's a pleasure. I'm going to talk to you today about telomere length. And telomere length has the potential to be a novel 21st century biomarker. So just to unpack that a little, I'm sure you're all familiar with the shape of a chromosome. Chromosomes take on this condensed X-shaped conformation during metaphase, and here DNA is condensed to form visible chromosomes, visible under the microscope. DNA is bound around protein histone units. And the reason for this is so that when cells divide, DNA can be accurately split between cells without losing any bits or having any mutations. As I'm sure you're also aware, the reason we're so interested in chromosomes is because these encode genes. Genes are a series of bases, A, G, C and T, and these base pairs make up the sequences that encode all the proteins that are the functional machinery of our cells. So I said that I was going to talk to you about a 21st century biomarker. So you might be saying to yourself, okay, you've told me what genes are, but what are telomeres? So telomeres are DNA protein complexes that are found at the ends of chromosomes. And what they do is protect functional regions that contain genes by capping the ends with what you could describe as a buffer sequence. So it's a little bit like the hem of a curtain. That's why I have a strange curtain picture up there. When the telomeres are completely worn away, that cell is destroyed. It's a signal to the body that that cell is old. What are telomeres? They consist of variable numbers of repetitive sequences, T, T, A, G, 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 in many copy numbers, and they're stabilised by a protein which has been aptly named shelterin. So what happens is that telomeres shorten with each cell division. In somatic cells, and somatic cells are your ordinary body cells. And this diagram depicts a cell shown in red, and as that divides, the telomeres shorten, and when they reach a critical shortened length, that cell is then destroyed. And this happens due to the way the telomeres are replicated each time the cell divides. So we have this important mechanism which tells the body how old our cells are. It gives them a sell-by date and it can get rid of them. This talk is about how we can look at this mechanism and understand when it's useful and when it goes wrong. We also have an enzyme which its job is to elongate telomeres. So telomere shortening is mitigated by an enzyme called telomerase. It doesn't really look like a shark, but I thought it was a nice diagram. It elongates proteins by adding our repeat sequence, T, T, A, G, 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 to the end of the existing telomeres. Now this enzyme is found expressed in cells that are rapidly dividing, like adult germ cells, fetal tissues which are growing rapidly, and also in cancer cells. And there's been great interest in looking at telomerase in cancer cells. So why do we particularly care about telomere length? Well, telomere length has been positively correlated with human lifespan. So genetic studies have looked at populations and individuals and determined that the length of their telomere is related to how long those people live. And so now we want to think, what can we do about that? So it appears, interestingly, that telomere length is influenced by stress. Stress, of course, is the great modern disease. And premature telomere shortening leads to premature aging of cells. So you're getting rid of cells that you want to keep, that should be kept, because you're wearing away their telomeres too quickly. Worryingly, it's been found that that's associated with socio-economic status. So are you poor? Your telomeres wear away more quickly. What do you eat? How much do you exercise? Bad things happening and being infected with things like HIV. So you might think that's a bit of a mysterious connection. How can these things happening in the wider world affect what's happening in our cells? Well, the connection appears to be oxidative damage. So the connection between the environment and shortening our telomeres appears to be mediated by oxidative stress. What is oxidative stress? It's an imbalance between the production of free radicals, highly reactive substances, and the ability of the body to remove their harmful effects. So we have lots of mechanisms to mop up free radicals. If our ability is reduced, they will stay in the cell and they will affect our telomere length. So, at the centre here, it says premature telomere attrition, that means telomere shortening. And feeding into that, we have socioeconomic status at the top. That's connected to physical activity. Often people who have lower socioeconomic status have less availability of going to the gym or cycling, running or so forth. And that's also related to diet. So once somebody has a poor diet, they're overweight, it's harder for them to exercise. Stressful life events often happen more to people with lower socioeconomic status because they don't have the money to mitigate problems that arise. And then there are factors that we have not got so much control, not got any control about, such as inheritance, heritable load. So how much of your telomere do you start off with due to your genetics? So all these things feed into telomere shortening, and they can result in accelerated biological aging and increased disease risk. So that can be immediate disease risk or disease developing later in life. About the inherited side of things, this is very interesting because although a variety of environmental factors result in human beings having individual variation in telomere length, recent research has showed there's a strong heritability, estimated at 70%. But what this means is if you start off with shorter telomeres, you're more likely to suffer adversely from accelerated cellular aging, i.e. other environmental stresses. So although we can't do anything about your heritable load, we can think about how to mitigate environmental circumstances for at-risk groups. Now, telomeres are rich in guanine, which is sensitive to oxidative damage. And oxidative damage can both alter shelterin binding, which was the protein that helped to stabilise our telomeres, and also inhibit telomerase activity. And remember, telomerase was the enzyme that elongated telomeres in early life. So telomere dysfunction, rather than just telomere shortening, might have a bigger effect on cellular aging. So the mechanisms of this are largely unknown, but obesity and things like smoking actually induce increased oxidative stress. And it's been shown that high levels of plasma oxidative stress are correlated with shorter telomeres in people with diabetes, with type 2 diabetes, which is of course associated with obesity. So these are chronic diseases that appear later in life. In addition, we have studies that show association with shortened telomeres between educational attainment, nutrition in infancy and childhood, exercise patterns, sleep patterns, socioeconomic factors, which I've said, racism, psychological stress, which I suppose racism would come under. So they've all been associated with accelerated telomere shortening and thus accelerated cellular aging. Just to explore the socio-economic status idea a little bit more. So low SES increases early morbidity and mortality. We know that. And the commonly used measures of SES are education, income, wealth, and occupational prestige. How can we connect these two things together, telomere length and occupational prestige, for example? Well, that's due to stress. Financial strain, hazardous home and work environments, and less access to psychosocial resources will all lead to more stress and more oxidative damage. A recent paper showed that African American children being exposed to stress, including low income and unstable family environment, were associated with reported telomere length at the age of 9. So this is impacting on children. Gene-environment interactions might differ significantly between ethnic groups, but we haven't done very many studies in other ethnicities. So if you start off with a shorter telomere, adverse life events will cause earlier cellular aging. Nutritional environment is key, and in women there's evidence that production of estrogen is also a risk factor for obesity, and that's related to length of reproductive years in life, and greater telomere and lower telomerase activity. But we can successfully intervene. A recent study showed that we could find an improvement in obesity after a 5-year Mediterranean diet intervention, and that gave rise to an increase in telomere length. So this slide is entitled Common Disease. So both shorter telomere length and its inverse, higher telomerase activity, have been associated with a variety of common diseases. And we're interested in common disease because common disease affects a lot of people. And these kind of diseases are breast and colon cancer, and that's associated with high expression of telomerase, diabetes, cardiovascular disease, hypertension, dementia, and blastocyst aneuploidy, which is where one has too many chromosomes in an early embryo. So telomere length impacts many, many individuals in our society. Now, as a scientist, we're interested in how to access cells in which we can measure telomere length. And if we do measure telomere length in those cells, are they representative of telomere length in cells in the liver, in the heart, in the lungs, sites of disease, for example? So you don't really want a doctor to be taking samples of your pancreas or your liver. You'd much prefer to be able to spit into a tube and look at your DNA in that. Luckily for us, buccal cells, which are cheek cells, skin epithelial, and things like synovial fluid, lymphocytes, which you can get from the blood, and vascular cell wall and umbilical artery, which you can obtain from a pregnant mother, are comparable. So although there are inter-tissue differences in telomere length, the relative telomere length is tissue independent. So if you take a norm and you say this person is 5% below, this person is 5% above, you can relate the tissues together. So that means that correlation found in different tissues can be used to predict the telomere length in other tissues. So that's good. Now, I said at the beginning of the talk that we wanted to use telomere length as a biomarker. And I've told you that once telomeres become critically short, DNA damage responses are activated, triggering aging in cells. Luckily for us, telomere length is easily and robustly measured via fluorescent PCR, and that can be done in lots of different cells, including cheek cells. So, I hope you also have some burning questions after my introduction to telomeres as a biomarker. What we're now interested in exploring is who is more or most susceptible to premature telomere shortening. Who are the at-risk group? What are the most significant risk factors for telomere shortening? Can we do anything about it? So telomere length may prove to be key in predicting the future health of children and young people and adults, in fact, and that is what a biomarker is. Can you look at something that's measurable and expressed before disease develops? And then can we use that information and intervene successfully? In response to these burning questions, we have an exciting research project going on at London Met, and our research looks at telomere length as biomarkers in Colombian children. Colombian Hispanics are an understudied population, and despite the fact that Colombia is a middle to high income country, there is a stark amount of socio-economic inequality, and that raises mortality from non-communicable diseases, which disproportionately affects the lower socio-economic status individuals. Biomarkers in these groups would be particularly useful to identify key factors in the development of disease risk. So we are examining this population to establish which childhood exposures have the greatest effect on telomere length and potentially on their future health. And of course we can extrapolate these results to other populations. So this is an innovative project measuring telomere length in Colombian Hispanic children aged 9, and we're going to compare them to their detailed life events, which includes a novel index of stress. So we've looked at these children, looked at their lives, looked at their home environment, whether their parents are in prison, divorced, whether they have a car, whether they have running water, what kind of diet they have, what access to exercise they have. And we're able to look at that, take that into an index of stress score and compare that to their telomere length. No other study has investigated all the targeted parameters in the population from the same age group that we are going to do, and we hope to identify factors associated with the telomere shortening in comparable lower income groups where poor nutrition, low physical activity and environmental stress in childhood is unfortunately common. So this work has the potential to be of high impact and also makes use of cross-discipline thinking. And I'm working with Dr. Wally in St George's, Dr. Butson in UCL and Professor Blakemore in Imperial College. So, in summary, we've described DNA structure in the nucleus, we've explained telomere structure, we've discussed the importance of telomere length, explored the impact of telomere length on human health, and also described exciting research being done to develop telomere length as a useful biomarker.